Hi friends, so in this video we are going to talk about glycolysis, which is the most important pathway which is required for the energy, and the products that are released are used in the further citric acid cycle and electron transport chain to release energy. And also when there is immediate need for energy, the glycolysis is the important pathway which provide energy. So please make sure to like, share and subscribe my channel. So what is glycolysis? It is also known as EMP pathway, Embden Meyerhof-Parnas pathway. So glycolysis, it derived from the Greek word glycos, which means sweet, and lysis, which means breakdown. What it means is the breakdown of the sweet, which means the breakdown of the glucose into different molecules. So what happens in glycolysis, the one molecule of glucose, it is break down to two molecules of pyruvate. So glucose is a 6 carbon molecule, and broken down to two molecules of pyruvate, which are 3 carbon molecule, and by a series of reaction which also need ATP, but then it releases ATP as well, and it requires in total 10 enzymes. Some of the steps are irreversible, which means they can go back and forward, but some of the reactions are irreversible, which means they can go only in the forward direction. And where does it happens? So glycolysis mostly happens in the cytosol of all the cells. So in the cytosol you have all the enzyme presence which are required for glycolysis, and the pyruvate that is released by the oxidation of glucose can enter, as I told you, in the citric acid cycle, the TCA cycle, to release more ATP molecules, or during the emergency need or during exercise it can also form lactate, which is the product where when it enters the anaerobic cycle. So the major pathway, as I have told you, the glucose, it can form pyruvate and by glycolysis, and the glucose can also form the ribose 5 phosphatase via the pentose phosphate pathway, and this is very important for the formation of nucleic acid. If there is excess of glucose, it can go to glycogen as the store in the store form, starch and sucrose as well. And if the glucose, it's also required for the formation of extracellular matrix and polysaccharides, so the glucose can go in this pathway as well. So as you can see, glucose has different pathway based on the needs of the cell and needs of the host. It can go to different pathways, and today we are going to talk about pathway of glucose to pyruvate. So the glycolysis happens, the breakdown happens in two phases. The first phase is called the preparatory phase, in which the cell is preparing itself for the glycolysis, and in this step the ATP is utilized. So if in this phase you spend energy to continue different enzyme reaction, and the another phase is payoff phase, where the ATP is released. So in the preparatory phase you spend some amount of ATP, like 2 molecules of ATP, but in the payoff phase you get 4 molecules of ATP. So in the end you are getting more energy from this pathway, and then this is the immediate source of energy whenever in need. So first we are going to talk about the preparatory phase, which consists of 5 reactions, and one of the first one is phosphorylation. So here you can see the 6 carbon molecule with the glucose, right, and it has the HOH, and this is the third carbon, as you can see here. And then by the action of the enzyme hexokinase, which is a kinase which phosphorylated, and it requires ATP, it gets converted to glucose 6 phosphatase. Like so, at the sixth carbon there is OH, and H is replaced by the phosphate group. So it forms the glucose 6 phosphate, and this enzyme, the reaction that occurs, it's the irreversible reaction, which means it can go only in the one direction. It is always forward. And now that we have glucose 6 phosphate, it enters into the different enzyme, which is the isomerization, What does that mean is the number of the carbon molecules remains the same, everything remains the same. It's just that there is some shift in the carbon numbers and the ring size. Like you can see here, the ring size is of like 6 carbon ring and 6 like yeah, and here it shifts into 5 carbon, and there is another CH2OH. So you can see from this shift from this ring there is formation of the another ring which is the same carbon number but the ring is different, and you form fructose 6 phosphate. So here you can see there is this shift of the carbon 2 to the place near the O, right, and one carbon move upward, and now it's called the fructose 6 phosphate. So this isomerization can go in both the direction, like fructose 6 phosphate can form glucose 6 phosphate or vice versa as well. Now that we have the fructose 6 phosphate, there is another phosphorylation on the first carbon. So before the phosphorylation was on the sixth carbon, now it's on the first carbon, and again we are utilizing the molecule of ATP. So now it's the second molecule. The first ATP molecule we used in the first reaction, and now we are using the second molecules of ATP, and the enzyme that is required is phosphofructokinase, and it adds the ATP to the first carbon. So here you can also see the enzyme, the reaction always occur in one direction, like in the forward reaction. So this reaction is irreversible, and so this enzyme is also the rate limiting enzyme as the hexokinases, and this controls the if the reaction will go in the forward direction, like to the formation of pyruvate, or not. So like there is a feedback mechanism that works with this enzyme, and from this phosphorylation we get two phosphates, and the compound, the molecule that is formed is called fructose 1,6 bisphosphate. And now we have the fructose 1,6 bisphosphate. Now we break down this molecule to three carbon molecules, 3 carbon molecules. So first, if like the breakdown occurs in the center, right, and then first you get 1 2 3 carbon CH2OP3, and there is some chain, like some shift in the bonds, and you get dihydroxyacetone phosphate. So this is a ketone. So what you get is dihydroxyacetone phosphate, and with the another carbon what we get is an aldehyde. So there is again a shift of one bond, and what we get is an aldehyde which is called glyceraldehyde 3 phosphate, and this 3 carbon glyceraldehyde 3 phosphate is the major source for the other pair of reaction. So now we have these, and this dihydroxyacetone phosphate converts into glyceraldehyde 3 phosphate again with the action of the triose phosphate isomerase, and in the end we get two molecules of glyceraldehyde 3 phosphate. So as I told you, the glyceraldehyde 3 phosphate is an important molecule for the further payoff reaction. So everything, like we start from the glucose and we get two molecules of glyceraldehyde 3 phosphate. So now we are finished with the preparatory phase. So the first 5 steps of the reaction are called preparatory, because if you see we use two molecules of ATP. So now we enter the payoff phase where we start getting the ATP back and more ATP as well, which starts with the glyceraldehyde 3 phosphate. So now that we have two molecules of glyceraldehyde 3 phosphate, and now we are talking about one molecule, and you can imagine the same for the another one. And with the action of the glyceraldehyde 3 phosphate dehydrogenase, there is an oxidation reaction which adds the phosphate group and also the H, and what we get is two phosphate molecules which is called 1,3 bisphosphate glycerate, right? So with one molecule of glucose we get two molecules of glyceraldehyde 3 phosphate, which means we are spending two molecules of NAD plus to get two molecules of 1,3 bisphosphate glycerate, right? So everything happens on the carbon which contain the aldehyde, like we add the phosphate group to the first carbon. So now we have the phosphate on the first carbon and the second carbon as well. And the second step, the next step is the release of energy. So now that we have two molecules of phosphate, what we get is these molecules of phosphate is released and added to ADP to form ATP by the action of enzyme phosphoglycerate kinase, and this reaction is the first reaction which is called the substrate level phosphorylation, which means the substrate here is 1,3-bis phosphate glycerate. This is like phosphorylating the ADP. So this is called substrate level phosphorylation because substrate is phosphorylating the ADP and releasing ATP. Which means because we will have two molecules of 1,3-bis phosphoglycerate, so we get already two molecules of ATP that we spent in the preparatory phase. So, and in what we get is two molecules of ATP and two molecules of 3-phosphoglycerate because first carbon phosphate is released and we are left with again 3-phosphoglycerate. And now there is some isomerization, some changes because the number of carbon remains the same. From this third carbon molecule, the phosphate moves to the second carbon and forms 2-phosphoglycerate. And with the help of the enzyme phosphoglycerate mutase. So now that we have 2-phosphoglycerate and so now there is dehydration, which means the release of water to form phosphoenol pyruvate. And here you can see the release of water from second carbon and the third carbon. And the water is released and what we get is phosphoenol pyruvate. This phosphoenol pyruvate is important because it can either go to form oxaloacetate, which can directly enter the citric acid cycle, or when we are moving into the reverse direction, like when we need to form the glucose, because suppose if the body is lack of glucose, we need to form glucose, which is the reverse of this. In that case, oxaloacetate can form phosphoenol pyruvate because the next reaction, the phosphoenol to pyruvate, this is a one step reaction, like this is irreversible. So oxaloacetate forms phosphoenol pyruvate to enter vice versa, like to enter back in this cycle to form glucose. So this phosphoenol pyruvate is very important. So now that we have phosphoenol pyruvate, another ATP is released because this phosphate is released to the ADP, and what we get is pyruvate alone, which has a like ketone group, enol group on the second carbon and carboxyl group on the first carbon, and ATP is released. So here we get again two molecules because we will have two molecules of this, so we get extra two molecules because if you see in the payoff phase we got 4 molecules of ATP but we spent only 2, so in total we get extra 2 molecules. And this pyruvate, it can be like isomerized, tautomerized to different forms like keto forms because these are important to enter different cycle. So in the end what we get is we spent 1 molecule of glucose, 2 molecules of ATP, 2 molecules of NAD+, and obviously 4 molecules of ADP because we needed the energy back, like the phosphorylation. And in the end what we get is 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. And in the overall, if we balanced it, because we spent 2 and we got 4, so in the end what we get in total is 2 molecules of ATP and 2 molecules of NADH, and which are enough because if you convert this NADH into ATP and it enters a electron transport chain, you will get enough energy, like the immediate energy if you require. So in the end we send like glucose can form 2 molecules of pyruvate, and where can this pyruvate can go? If the conditions are aerobic and everything is fine, it can enter the TCA cycle, citric acid cycle, to form energy and releases carbon dioxide. Under anaerobic condition like stress and the muscle usage, it can form lactate as well, which can then enter the cycle and can release energy. And for the fermentation, like for hypoxic condition, it can also undergo the fermentation and can also form ethanol. So the important which happens, which provides you more energy, is this pathway pyruvate to acetyl coenzyme A and the citric acid cycle, which provides you 36 molecules of ATP. So I hope it's clear, and if you have any doubts, please let me know in the comment section, and please like, share and subscribe my channel. Thank you.